Thank you everyone for joining today. So I'm happy to share today our recent work where we aim at delineating the functional patterns in global gut microbiomes. So as you all know, microbiomes are a complex community of microorganisms, and functional interaction between macros will define the community functions and behavior. Now, today I will focus on the human gut microbiome, and we know that the human gut microbiome goes through several compositional transitions until reaching a somewhat stable state at the adult stage. So I will focus on the adult gut microbiome. And what's interesting with the adult gut microbiome, although we all reach this relatively stable state, is that we identify a lot of inter-individual variation in the composition of the gut microbiome. That being said, even if we have those inter-individual variation, it has also been shown that if you look at the gut microbiome across the global population, those profiles can be classified into 3 different large configurations that were early on named enterotypes. And those configurations are characterized by the predominance of a specific taxa. In addition, interestingly, those major compositional configurations were also linked to the host status, health status, diet, and specific metabolic and immunological traits. So this is, I think, very interesting, but the thing is that many past definitions focused on the composition rather than function. And we believe that characterizing the functional potential of microbiome rather than composition is important, as recent studies show that the functional characteristics associate with the microbiome stability and resilience. So our research aims to define this functional variability to see if we can identify such a similar type of configuration that emerge when we look at the global healthy adult gut microbiome, and investigate the interplay between those functional configuration, compositional configurations, and also its relations to gut microbiome stability, resilience, and disease. So to tackle this problem, this question, a PhD student in the lab, Mohammed, has been using deep archetypal analysis. So this is a deep learning framework combined with archetypal analysis that has been used in various type fields successfully, and including in computer vision. And I added this example just so you have an idea of what I mean by archetypes, and also how the different profiles will be kind of mapped into this archetypal space. Here, it's defined by 3 archetypes. And so each dot is a functional or like a gut microbiome profiles. And the dots and each profile will be given a score. And the score is the strength of how it relates to those extreme states, those archetypes. So for example, those profiles will have a very strong score and are close to this extreme profile defined by archetype 2. Meanwhile, the profiles in the middle are kind of combinations of the 3 archetypes. And so here, for a bit of a more visual example, you have a model that classifies facial expressions. And basically, the archetypes that are identified here are extreme expression of happiness, anger, and surprise. And then the model can order or classify the other facial expression as a mix of those different facial expression. So it's a deep learning approach. So for this, and we also wanted to look at as I mentioned, the variability across the global healthy gut microbiomes. So we curated about 11,000 whole-genome metagenomic profiles publicly available across 76 studies in 31 countries. Unfortunately, especially when you start to work with whole-genome metagenomics instead of 16S, we tend to have an enrichment of profile from the global north and like global south profiles, but we are keeping an eye out to include as many global south data sets as possible. We then profile all those metagenomics profile following this pipeline using Kraken2, Bracken, and the HumGut database to identify the species in the population to then process them with human3 so that we have relative abundance of metabolic pathways. And at the end, we had about 10,000 samples after filtration of samples and pathways, and relative abundance across 436 of those pathways. So it's an unsupervised problem, so we don't really know how many archetypes are there, but here we identify 3 archetypes that were able to capture this variability. And you can see that the samples tend to be in this type of transition states between the archetype 3 and archetype 1. In contrast, samples that are close to archetype 2 tend to be a little bit isolated. You don't have so much transition from across those functional states. The other thing we observe is that the archetypes kind of capture functional diversity. So the samples that are closest to this extreme state, the archetype 2 state, tended also to have a more diverse functional profile, so more pathways detected in those samples. And this was not matched with an increased relative diversity of species. So now, what do those archetypes mean? So we link now back the pathways to define those archetypes and see what pathways are more highly expressed for the samples that are closer to those extreme states, extreme functional states. So here the samples are in columns and pathways are in rows, and it shows the top 20 pathway defining those functional archetypes. And when you look at the processes of this pathway, you can see different metabolic processes associated with the archetypes. So archetype 1 is characterized by high potential for carbohydrate sugar metabolism and biosynthesis of branched-chain amino acids as well as cell wall component. Unlike archetype 1, archetype 2 prioritize fatty acid utilization and energy metabolism and nucleotide biosynthesis. And if you remember, archetype 2 was also the one that was more functionally diverse. So we hypothesize that this diversity is probably allowed due to this high energy potential. Finally, archetype 3 may favor microbes that with enhanced nitrogen metabolism capabilities, particularly in processing nitrogen compounds, including amino acid, through the urea cycle and related pathways. So how does it relate to the compositional, the well-known compositional archetypes? So there are some association. It's kind of expected. Again, here we look at the DNA, that the DNA pool present in the population. So there are some relationship with the known enterotypes, but it's not a one-to-one relationship. So all the samples that were classified are more likely to be prevotella, tended to be all from the archetype 1, but you also have a lot of samples that are closer to the archetype 1 that belongs to the Firmicutes classification here. The archetype 2 tended to be mostly Firmicutes, but Firmicutes is not only archetype 2. And finally, there is a relationship between an enrichment of Bacteroides samples related to the archetype 3. So again, like we wanted to also investigate how this archetypal analysis was providing information about the stability. So for this, we selected studies with repeated measurements, and we have profiles for 666 individuals. And we looked at how they were, this functional profile was changing across the visits. And what we observe is that if a profile is classified very closely to one of those extreme states, it tended to be very stable and stay in this state. In the meantime, in contrast, like when the samples were a bit more in the middle of this map and belonged to a mix of archetypes, then they tended to be less stable. So then we looked at disease samples, and we looked at samples obtained from IBD patients, but also colorectal cancer and type 2 diabetes. Here I'll just present a few results from the IBD studies, and we can see that the IBD patients are not exactly distributed the same way in the archetypal space compared to healthy controls. And you can see there are an enrichment of samples that are closer to the archetype 2. And so, but we still have a lot of IBD samples that fall in this other space. So when we do compare case control studies and compare the IBD patient, we thought that it would be interesting to compare only, for example, the IBD samples belonging to close to archetype 3 compared to the healthy that were in the same type of configuration. And we did so if we don't do that, we combine all the IBD patients together and compare to healthy, and we identify functional pathway differentially enriched, but it's also very much associated also with the, like I mentioned, the type 2 archetype score. So instead, when you look at the stratified analysis and looked only at the type 1 dominant samples for comparing IBD versus healthy, or, for example, the type 3 dominant samples, IBD versus L3, we identify different functional pathway differentially enriched into these two groups. And so this is very interesting because it can be that the different IBD archetype-specific signature may reflect differences in the disease subtypes and clinical presentation in disease stage. IBD is a very heterogeneous group of disease, but it also could inform on a specific targeted therapy that will work better when this is looked into an archetypal specific way. So just a short summary, we have characterized the functional diversity of healthy gut microbiomes using defining three functional archetypes. It associates with microbiome stability. It can potentially confound disease association, but also reveal therapeutic targets that will be more appropriate once we stratify the samples. And this deep learning framework is very interesting and provide this robust characterization of microbiome functional systems. And I want to thank the team and the founding, and I'll be happy to answer any question.